Okay. Is anybody in here undergoing cell division right now? What do you think? Someone really? Yeah. Anybody who's not undergoing cell division right now? If you're not, you're dead. Just want you to know that, so you better get with it. Okay. We in Symbio, the mitosis was just turned in, and that's what cell division is all about. That's the actual mechanism behind it. So what you're doing with cell division is you're making daughter cells that are identical to the parent cell. Has nothing to do with reproduction, unless you're a single cell organism. So what you're doing is you're making clones. Now, of course, there's always a chance of mutation, and in fact, that's what happens with cancer. You need many mutations, and then that gives rise to cells that are uncontrolled in terms of cell cycle.
So like there's mitosis, right?
Yeah.
So cancer cells do
Mitosis. And then meiosis for reproduction.
Alright.
Right, exactly. Now meiosis obviously arose from mitosis, but it's subsexual reproduction, so you don't necessarily need it. Okay, so for cell division to happen, you need to replicate the DNA. That means we have 23 pairs of chromosomes. 22 are identical. Those are called autosomes. And then there's one pair of sex chromosomes, X and Y. Females have 2 X's. Males have an X and a Y. And so males are actually the heterogametic sex, and we'll talk more about that. But that's why males generally show more bad mutations that have an effect on fitness than females do. Because oftentimes there's a rescue effect. So with normal chromosomes, there's 2 of each. So if you have a bad mutation in one, there's usually a rescue from the other one. But with an X and a Y, if there's a mutation on the X, for males there's no recovery. So that's why there's a whole bunch of sex-linked problems. But we'll get to that much later. One of the things that happens is that we replicate the DNA, and the first thing that actually happens is there's condensed DNA is condensed into actual. You can actually see the chromosomes. Otherwise, normally in a cell in the nucleus, you can't see the DNA. It's all spread out, and then the DNA is completely copied, so that you have 2 full copies of the genome, and that way the original cell and the daughter cell now both have their own copies. So this is kind of what uncondensed DNA looks like. It's actually partially condensed. If it was all condensed in chromosomes, it wouldn't be able to function properly. One of the reasons that it's able to function is to spread out, and certain sites are then able to actually produce proteins. Okay. So this is what condensed chromosomes actually look like. We each have 2 copies. So okay. One from mom, one from dad. The chromosomes have a long arm and a short arm, as you can see. And then there's certain things that are, so where they're actually attached, right here, it's called the centromere. Each of these chromosomes is a chromatid, sister chromatids, okay? So the full chromosome has a copy, one from mom, one from dad. And then each individual from mom or from dad is a sister chromatid. Now, there's something called the cell cycle. Most cells are in the G1 phase for most of their lives. Some cells never get out of the G1 phase, like neurons. Others go through the cell cycle to replicate. And then there's something called the S phase, which is interphase. The whole thing actually is interphase. So this all is interphase. Okay? And then, of course, we have mitosis. Okay. So there's cell growth and function. Then there's a signal, at which point all the DNA is copied completely. So you have now 2 complete copies of the genome. And then there's more cell growth and preparation for division, and then you finally go into mitosis. So, I want you to remember what the actual cell cycle is and what happens in each. Okay. So, this is a real quick look at mitosis. So mitosis is this part right up here. And here we have interphase, where the DNA is being copied, the cells are growing, getting enough resources to actually divide. And then mitosis starts from here. and goes through here. And the basic steps are, it's called prophase, where the DNA is condensed into chromosomes. Metaphase, where all the DNA lines up, what's called the metaphase plate. And then there's these spindle fibers that connect to the DNA. Pull them apart, because remember there's now 2 copies of each, so that each cell has a complete copy of the genome. And then it divides in 2. So, same sort of thing again. This is in a plant cell. Plant cells are, a lot of them are large, it's easy to see. So it's prophase, metaphase, anaphase, where they're pulled apart, and then telophase, where you actually have 2 daughter cells. Same sort of thing here. Now, why do cells divide? Three things. First of all, the body might need to make cells over and over again, like red blood cells. Billions are produced at all times from the stem cells in the bone marrow. And so you need continual replication. Skin cells are the same way. And then, of course, there's tissue renewal, either because cells wear out, or they get damaged, or something else like that happens. Let's see. So these are right here. This is blood cells. The red ones are the red blood cells. And then these are lymphocytes, part of the immune system. And then in terms of how often it happens, cells have different lifespans. So, skin cells, they're replaced daily or over a few days. Blood cells, over a month or months. Muscle cells and cell organs, depending on what you're doing, like with muscle cells, you can break them down when you're working out. So if you want to build up your muscles. But a lot of the cells and muscles and organs, they're really not, they last a long time. Now, of course, they also divide because they're lonely. And then, occasionally, cells go what we call rogue. And that has to do with cancer. Now, of course, they're different cells, have different genes being turned on in them. And this is what's called differentiation that we probably have already gone over. So different kinds of cells. They arise from stem cells, which we'll get to later, whether they're adult stem cells or embryonic stem cells, that sort of thing. Okay. Now, occasionally, when cells divide, they get signals to divide. It's a very tightly controlled, or it should be, a tightly controlled process. But sometimes things go a little haywire. There are certain genes that, if they're turned on, they're called proto-oncogenes. Oncogenes are genes that perhaps cause cancer if they get a mutation in them. Now, it's a kind of a misnomer that they're actually cancer-producing genes. They're not. They're cells that are doing normal function, but if there's a mutation in them, let's say there's a mutation and they keep reproducing uncontrollably, then they're actually called oncogenes, and they've actually the start of cancer. So it's pretty much like piracy. Now, cancer is an outcome of having multiple cells making up an organism. So if you're just a single-celled organism, the actual stuff that's, let's say, could cause cancer, let's say a mutation, could happen in a single cell, like a paramecium, any sort of single cell eukaryote. But, and let's say one of those cells might start dividing because of mutation, even though it's really not ready. In other words, it doesn't have the resources to actually make a copy of itself. And so if a cell did that, it would probably die. The thing is, we have multiple trillions of cells in our body, and the cells essentially, since they all have the same genome, they're all related to each other. And in a sense, what cells do is they give up their own sovereignty. In other words, they're controlled so that they just don't go willy-nilly off dividing and reproducing. So reproduction, remember in evolution, fitness, that sort of stuff? Well, individual cells give up their own fitness for, I won't say the good of the body, but it's very tightly controlled. It's more like a police action than anything else. In other words, if a cell starts getting what we call hinky and acting abnormally, there's a whole system out there to stop it from doing that. That's part of the whole cell cycle that we were looking at. But occasionally, there's enough mutations that this control breaks down. And then you end up with cells that are reproducing at the expense of all the other cells in the body. Okay. Now, as I said, it's a very controlled process, cell division. And it actually, because of all these kind of backup systems, anytime a cell has something funky going on with it, it usually gets recognized by tumor suppressor system. There's a whole bunch of them. P53, BRCA 1 and 2, that's the one that's usually associated with breast cancer. Other parts of the system are when cells replicate, sometimes there's a mutation, and there's these biochemicals that go along And if there is a mutation, they go on and change it. Or if they can't do that, then it stops the cell cycle, and that's the end of it for that cell. Okay. So, most cancer has nothing to do with inheritance. In other words, inheriting a specific kind of propensity to cancer that you might get from your parents. There are cancers like this, but most of them are, what are they called? They just happen spontaneously. And that's why there's a system for making sure that if it does happen, the cell gets stopped in the cell cycle and doesn't keep dividing further. Cancer is a yes.
How about the breast cancer? You know, how like if your grandma has it, she passed on to your mother, or else the next generation.
Yeah.
How do you think?
Okay. So a good example is the cancer that women get for breast cancer, and also ovarian cancer, and it's also, it turns out it's also a part of prostate cancer too. It's a tumor suppressor gene. That means when it comes across, the protein comes across a cell that is not acting correctly, it stops that cell from dividing. Well, if you have a mutation in that gene, it means the tumor suppressor part of it is not working. So if it comes across a cell, it still keeps dividing because that system is not in place and working anymore. If you have a mutation in one of those genes, they can be passed on to your offspring. Now, like for the BRCH gene, it's about, there's something called penetrance, how often it actually shows up and, let's say, causes cancer. So for breast cancer, it's with the BRCA gene, it's about 20 to up to 80% of the time. It will lead eventually in a person's lifetime to cancer. There's others like there's a cancer of the eye where the penetrance is up to between 90 and 100%. So almost everybody who inherits that from a parent will end up with an eye tumor. Okay, so it depends on, but that's basically how it is. Now most cancers are not because of that. It's because of during their lifetime as they divide, a mutation happens, and then perhaps another mutation happens, and eventually they start controlling, they divide uncontrollably. That's a mutation during mitosis, not during meiosis. Okay? That's the difference. So there are cancers that are genetic, where they're passed on from one generation to the next, but not most. And some of the mutations that are passed on, they don't necessarily lead automatically to producing cancer cells. So, in other words, you have a tumor suppressor gene that's got a bad mutation, it's not doing its job. That means that you have kind of a head start in perhaps developing cancer. Okay. So, in a sense, cancer is always genetic. Because remember, during mitosis, cells are dividing, all their genes are copied, So you have two complements of the full genome, and then they divide into two daughter cells. But in terms of genetic being passed on from parents to offspring, that only happens in what are called germline cells. Those are the cells that produce eggs and sperm, and that's meiosis. We're going to get to that later, but for now, we're not. So when we often say genetic, it's passed from one generation to the next, but ultimately, cancer is a genetic disease. You just might have mutations, though, in the cells that aren't passed on from one generation to the next. So I go out in the sun. I get radiation damage from the sun. That causes perhaps some mutations, and I end up with a skin cancer. That's not passed on from one generation to the next. That's just normal cell division gone haywire. Is it environmental? Yes. In the sense that all kinds of different things can happen. Radiation from the sun, that's environmental. You can have carcinogens or mutagens that cause mutations or cause cancer to develop from environmental factors, like smoking. In fact, they've, in the last couple of years, they've actually shown step by step, in terms of mutations, how lung cancer gets going from getting damage from nicotine and all the other stuff that's in cigarettes. You can also get cancer from certain viruses. So a virus inserts its DNA into the cell's DNA to make copies of itself. Well, you can understand that that might mess things up and cause those cells, if they're not destroyed, to start copying uncontrollably. Okay? One of the best known ones is HPV, human papillomavirus. Causes cervical cancer. It also causes mouth and throat cancer if you engage in oral sex. Is cancer contagious? Like, if someone comes along and you rub them against someone, can they pass it on to you? Well, no. But unless you're a dog or you're a Tasmanian devil, which is a kind of marsupial. And then there actually are contagious cancers. The kinds that dogs have, they might pass it on. They pass cancer cells onto each other when, let's say, they're licking each other. The Tasmanian devils, they have absolutely wild, violent sex, where they're biting each other constantly, and that's how that gets passed on. But there's no human cancers like that at all. Oh, let's see, a lot of clams also have cancers that are actually contagious. Usually, our immune system, including the tumor suppressor system, is what protects us from cancer. In other words, it recognizes cells that start acting inappropriately, whether it's because of damage to them, it's causing mutations, whether it's from a virus, whether it's a mutation that's been inherited. Normally, your immune system protects you. And then is the treatment worse than the disease? Well, in many cases, yes. Okay. Now remember, we were talking about mutations in DNA, not only do they turn genes on and off, but that they actually, it's more like a dimmer switch, actually. So it's not just on or off, but the amount of gene product that's actually being made. In other words, the amount of protein that's actually being made. Okay? But let's say it's an on-off sort of switch. If a gene gets turned on and it stays on, that can cause a problem. It can also, with a mutation, alter the function of a protein. So normally it makes a protein with a certain amino acid configuration, and if you get a substitution in a certain area, you'll end up with a protein that's, let's say, not functioning properly. Many times it'll have no effect, but sometimes it does. Okay. So what are genes that encode proteins that promote cell division when needed? Starts with an O. Anybody? Oncogenes. Okay. And I don't like the term oncogenes because it means that it sounds like it's actually made to turn into cancer, and it's not. But if you have genes that have a mutation that can then lead to uncontrollable cell growth and replication, that's when you can have cancer. So they're called proto-oncogenes. Proto means they're not doing it right now. They're not causing cancer. But if there is a mutation and they're jammed on, then they possibly will cause cancer. So which type of mutation do you think would cause the most trouble? A mutation that inactivates a proto-oncogene or one that activates it? Anybody? Huh? Yeah, exactly. So in other words, that gene has to be expressed. Okay. And then if it's actually doing that, being expressed and then replicating uncontrollably, that's now an oncogene. Okay. Now, tumor suppressor genes. There's a whole bunch of different kinds. Some are expressed more often in certain tissues than others. When a cell starts exhibiting perhaps certain things on its outer shell that show that it's starting to replicate uncontrollably or there's something wrong with it, there's these tumor suppressor genes that are expressed and produce certain proteins. There are other genes that when a cell replicates, they make sure that when the DNA is actually replicated, that everything happens so there's a proper complement. On the complementary strand, an A will have a T. A G will have a C. There can be, that's part of the tumor suppressor system. So if there's a problem there, it'll either fix that nucleotide, or it'll stop the whole division. Now, during the cell cycle, there are these checkpoints that a cell goes through. To pass on to the next phase of cell replication, you actually have to pass through the checkpoint where everything is all copacetic. Okay? So, the G1 checkpoint, first of all, a cell needs a signal to start dividing. Has that cell actually received that signal or not? If it turns out that a cell starts dividing, and, but it comes to that checkpoint and it hasn't received the proper signal, it will stop. Are growth factors present? So when the cell divides, making copies of the cell, you're actually making cells are growing. So let's say during development, you start with a zygote and it starts dividing. And it differentiates depending on which genes are turned on and which genes are turned off and how much they're turned on. There are these things called growth factors that actually enhance that. So like there's, when cells divide, oftentimes they need more oxygen and nutrients, and so they also start the production of blood vessels to carry blood and oxygen to the new cells. Okay, so there's a growth factor for that. Is its cell large enough? In other words, does it have enough resources to actually survive making a copy of itself? And so there's enough resources. Now, there's also a checkpoint at the end of the G2 phase. So, at which phase is the DNA copied? G1, S, or G2? Huh? S, exactly. G1 is the normal phase that a cell is in. It's functioning properly. It's making proteins, doing what it's supposed to do. G2, it's already made a complete copy of the genome, but now that cell also, it has to basically duplicate everything in the original daughter cell. That takes a lot of resources, including nutrients and energy and all that kind of stuff. So, at the G2 phase, after the S, was the DNA replicated correctly? In other words, were there any mistakes or not? If there were enough mistakes, it'll stop there, and it will not proceed to mitosis. Again, is the cell large enough and does it have enough nutrients? Tumor suppressor genes are involved in all these different checkpoints. And then there's also a checkpoint during mitosis where are all the chromosomes attached to what are called microtubules. We'll get into that when we get into a little bit more detail about mitosis. Okay. So, mutations that activate or inactivate tumor suppressor genes are dangerous. Which ones are dangerous? The one, what? Inactivate, right. In other words, the tumor suppressor gene, to do its job, it has to recognize a cell that's not acting appropriately. And then that will normally happen when this tumor suppressor gene has been inactivated. In other words, you get a copy of the protein that's a bad copy, and it's not doing, it doesn't have the proper amino acids, so it can't do its normal function. Okay. Now, BRCA1 and 2 are the tumor suppressor genes that are the basis for a lot of breast cancers. And women who have a mutation in them have a much higher risk of actually developing breast cancer. A lot of women who discover that they have this mutation decide they're going to have both breasts removed because they think the risk is too high. That's what Angelina Jolie did. It's not necessarily that it's absolutely going to happen. As I say, there's, depending on how much of a mutation there is in the BRC tumor suppressor gene, there's anywhere from between 20 and 80% chance that during a woman's lifetime she'll develop breast cancer and also ovarian cancer. And for men, there's some evidence that it controls prostate cancer as well. Now, when a tumor suppressor gene comes across a cell that's doing something wrong, what it'll do is it'll actually cause that cell to self-destruct. In other words, it'll tell the cell, here's a gun, put it to your head and shoot yourself. That's called apoptosis. Okay. Apoptosis. So that's when a cell gets a signal from a tumor suppressor protein that it's blown it. And it's time for it to kill itself. So here we have a normal cell, and here we have a cell that's actually destroying itself from the inside out. Now, if a tumor suppressor gene is malfunctioning, that means if mutations in other genes accumulate over time, and they are happening, normally they would be suppressed by the tumor suppressor gene, but if it's not doing its job, then those cells that do have a mutation, instead of being stopped in their tracks, keep replicating. Okay. Now, the little video that we looked at, it showed what happens when there's uncontrolled cell division. Usually you get a tumor, unless it's a blood cancer or something like that, like leukemia. Now, most tumors are not cancerous, they're benign. In other words, they don't invade adjacent cell tissue, nor do they travel around the body. It doesn't mean that they're not dangerous. There are a lot of tumors, like a pituitary tumor in the brain, that's benign, but it's still slowly growing, and eventually it can cause serious problems and even death. But for the most part, a benign tumor is usually much more treatable and not nearly as dangerous. Okay. So there's something that causes a benign tumor not to invade adjacent tissue. And that is called contact inhibition. When cells lose contact inhibition, they come up against another cell or other tissue, they lose it, they can start growing into that tissue. That's invasive, that's malignant. That's cancer. Cancer, not all cancers are the same. Some are very slow growing and are unlikely to actually cause someone to die from it, even though they may have it for a long time. So there are some prostate cancers that are very slow growing, and usually they don't treat those. They monitor them, but they don't treat them. And eventually the person is going to die from something else before they're going to actually die from, let's say, that kind of prostate cancer. There's other kinds of prostate cancer that are invasive and dangerous. Okay? So when a cell is invasive, that means it starts growing into adjacent tissue. That means it's a malignant cell and it has lost contact inhibition. So this is kind of a progression here. We have a tumor that starts forming. It could be a benign tumor, and that's as far as it's going to go. Okay? Or it can be a tumor that starts invading adjacent tissue, and then it's malignant. And what has it lost? Allowing it to invade, what is it called? Starts with a C.
Contact inhibition.
Inhibition, right. Okay. Actually. Now, there's also something else that's going on. That little video mentioned when cancer can spread from one place to another. Now, normally cells don't move around. Let's say a muscle cell. It stays in place. It's actually got something called anchorage dependence. That means it's not going to just get up and start wandering around or getting into the bloodstream or the lymph system. But when it loses anchorage dependence, that means that it can start traveling. So not all cancers are metastatic. Some remain in place, they invade adjacent tissue, but they don't travel anywhere. Whereas others can travel extensively. So lung cancers, breast cancers, pancreatic cancers, all of those are actually metastatic. Not only do they invade surrounding tissue, but they can generate tumors in the brain, in other organs. So, we have malignant. If it's non-metastatic, it means that it's invading adjacent tissue, but it's not traveling anywhere. And then metastatic. So this is the kind of progression. Okay. Actually, normal cell division. We make daughter cells that are identical to the original cell. They go through all the checkpoints fine. mitosis, all that whole bit. And then you've got normal cells that are being replaced. Now, potentially cancerous cells, you have unregulated cell division for whatever the reason. This is a benign tumor, and this is up here is a malignant tumor, a cancerous tumor. Okay? Okay. And then benign, no effect on surrounding tissue. Malignant, yes. And then finally, metastatic. Does a malignant tumor generate cells that are able to travel all throughout the body? Okay. A substance that actually causes a mutation that causes uncontrolled cell division is called a carcinogen. Cigarette smoke contains a whole bunch of substances that damage cells and that actually start the cancerous process. And in fact, I recently read an article about how it actually does that, the actual molecular steps and which biochemical pathways are interrupted. Anybody remember what free radicals are? Or anybody remember what will tends to stop free radicals? Antioxidants. Yeah. Antibiotics. Yeah. It's anti. It's anti, right? So it's good enough. Okay. So in other words, if you still want to smoke, eat lots of blueberries. Right? That means you can smoke all you want, but you have to eat lots of... No, don't do that. Every time you want to smoke, I would just eat blueberries. Okay? And not smoke. Or, if you have a significant other, every time you want to smoke, I would just kiss them. Kissing is much more pleasant than smoking, as far as I'm concerned. But anyway, that's just me. Okay. Okay. Now, so it's not just lung cancer, it's also mouth cancer, throat cancer, esophageal cancer, a bunch of different kinds of cancer, because basically, I mean, you can swallow a lot of these chemicals, and stomach cancer, things like that. Okay, so carcinogens, UV light, X-rays, ionizing radiation, tobacco smoke, chemicals, certain plants make chemicals that actually can cause cancers, like hydrogen cyanide is a common chemical found in seeds to protect it from getting eaten. That can also actually cause cancer. There are also things that are just called mutagens. They just cause mutations. They're not necessarily carcinogens, but some are. A poor diet. So I guess if you eat double all the time from morning through night, you have a higher incidence possibly of stomach or colon cancer. And then there's genetic predisposition. There's also a few viruses. Now, exercise is good because it actually keeps your immune system healthier and doing its job. Unfortunately, as you get older, your immune system starts falling down on the job. And so, even though you might hear about people getting cancer at a younger age, even childhood, for the most part, cancer is an old person's disease. Their risk of developing cancer increases with age. It's a real bummer. I'm against it. The only way to escape it is to die before you get cancer. So, if in fact, if you remember, you can prevent cancer by not wearing your seatbelt. Then you can get in a car wreck.
Oh my goodness.
And you will die, and you will prevent cancer. Okay? Okay, so this is kind of where we left off, or it is. What's a tumor suppressor gene? Anybody? Okay, you right here. I want you to answer. What's the tumor suppressor gene?
Oh, suppresses something. Oh, thanks.
Yeah.
Makes the proteins that stop cell division.
What?
Makes the protein that stops cell division.
Uh-huh.
Is it BRC?
What does it actually do? Not only does it stop it, but what does it do to the cell? Huh?
Was it something else?
See, I was right.
Yeah, but that's another word.
But that's what makes it, and that's what it's called.
What is it called?
Apoptosis.
Apoptosis, exactly. Excellent note-taking. So, tumor suppressor genes, they make a protein that recognizes a cell that's getting a little hinky. And whether it's been invaded by a virus or something else has gone wrong, and it basically gives a gun to the cell and says, it's time to commit suicide, and it does. It blows its brains out. What's the difference between a malignant cell and a benign cell?
Benign means it's non-cancerous and it's not going to hurt you. Malignant is
Okay, but
the opposite.
specifically.
It invades tissues and
Yeah.
hurts them.
Exactly. So a malignant cell invades adjacent tissue. Now, normally what happens is for all cells, when they come up against adjacent tissue, they stop dividing. Okay? And that's called contact inhibition. With malignant cells, they've lost contact inhibition, they keep growing. So, cancer cells, well, let's put it this way. When an embryo is dividing, cells need oxygen and nutrients. And what they do is there are these growth factors that, in fact, start making blood vessels to bring blood to those new cells. So that's what all cells do. It's called angiogenesis. That means the creation of new blood vessels. And cancer does the same thing. So cancer, in fact, in some senses, because it's dividing all the time, needs a tremendous amount of energy in the form of glucose and oxygen. And so normally what we see is a lot of those nutrients with cancer patients, they're all going towards the cancer. And often patients end up losing a lot of weight because all that stuff is going to the cancer cells instead of the actual patient. In fact, some of the drugs that are used to treat cancer are to stop angiogenesis from happening because usually, for the most part, the body's already got all its circulatory system actually working. And so the only cells that are going to be needing extra blood and oxygen and all that are going to be cancer. So that's one of the ways that we actually treat some cancer cells. Now there's another thing. Okay, we talked about tumor suppressors, of course. One of their functions is to stop cancer from happening. Well, tumor suppressor genes are all part of the immune system. And remember, originally, these cancer cells are normal cells that are part of our body that then, because of mistakes, start reproducing and replicating. One of the things that cancer actually does, it creates immunosuppressive environments around their own cancer cells, so the body's immune system is not going to be attacking the body's own cells. And so instead of an immune system coming along and with those proteins and making those cells commit apoptosis, they're protected from that. So what they do is they stop the immune system from attacking cancer cells. And there's a whole bunch of different ways to do it. There's immunosuppressive cells, there's immunosuppressive molecules. So immune system, suppressive, to suppress. And so I just want you to make sure you understand this. In fact, that was one of the big questions. How come the immune system was not actually stopping this from happening? And when they initially started to try to get the immune system going to recognize cancer cells and destroy it, it really wasn't working. Until recently. And now you've probably heard about immune therapy. We'll get to in more detail a little bit later. But immune therapy is a number of different methods for getting the immune system to actually attack cancer cells. Okay. Now, cancer, the current model is that cancer progression requires multiple mutations for cancer to actually start. One mutation will usually not turn a cell cancerous or malignant. It takes multiple ones. In fact, if we think about tumors, and as these replications are happening, there's more mutations that are happening. And so you can actually look at a tumor and sequence it and see how it's actually progressing in terms of the mutations it's actually acquiring. So oftentimes, an early malignant cell may just be invasive, but not metastatic. Anybody remember what metastatic is? How about, anybody like to go on vacation? Yeah. So, what is it?
It's spreading to different regions of the body.
Right, exactly. So, metastatic means cancer cells actually take off and go on vacations in the brain, various organs, and those are secondary tumors. Oftentimes, that ability doesn't start initially with the first invasive cancer cells. It happens later on. And so, because of more mutations. So this model that we have is called the multiple mutations or multiple hit model for cancer, where it takes many mutations and cancer cells actually, the population of cancer cells actually evolves over time. So here's melanoma. Most skin cancers, by the way, are very slow growing. They don't cause a problem, but melanoma does. And once it actually starts invading surrounding tissue, it metastasizes very quickly, and you end up with tumors all over the place. And so for like stage 3 and 4 melanoma, the prognosis is really poor until recently because of immunotherapy. So they're able to treat it now more successfully than they were in the past. So anytime you get funny looking spots on your skin, you should have those looked at. Oftentimes, obviously, it's because of solar radiation hanging out in the sun. Anybody like to just sunbathe? Yeah, exactly. It's great. You're just out there. Your hair turns a little, blonde. You're nice and bronze. That's great, as far as I'm concerned. I'd rather look like that than actually, than not look like that and get melanoma, right? So I think you should all do that. Now, in terms of cancer detection, one way they do it, there's not a lot of different ways for different kinds of cancer. Of course, one way is to do a biopsy. So like that skin cancer, they would slice off a little bit, take it to the lab and look at it to see, in fact, if it's, let's say, melanoma or some other kind of skin cancer like squamous cell skin cancer. You can also get biopsies for, let's say, liver. You just poke a needle in there, and it's kind of brutal. So that's one way. Also, some cancers, there are biochemicals that are released that indicate there's cancer in the system. So prostate cancer, there's something called PSA. Now you can get higher PSA levels just from a prostate gland getting bigger and bigger as a man ages. But if you see a spike in PSA levels, and it's one of the standard things they actually measure when you ever get a blood test, if they see a spike in that, they'll then go in and usually do biopsy of the prostate gland. And there's other things. Cancer causes, various different kinds of cancer cause many different strange symptoms. So let's say we've got a liver cell that's cancerous. Oftentimes with a liver cancer or a pancreatic cancer, something like that, those cells are still making hormones. And so there's an overabundance of these hormones circulating, and they're creating additional symptoms. So someone thinks they're fine, but they're starting to get strange symptoms that, let's say their eyes are not working as well, something like that. And so some of these symptoms are actually an indication that there's cancer somewhere in the body. But still, there's not a blood test that you can do for all these different kinds of cancer. And it's unfortunate. Steve Jobs. So, he doesn't look like it, but Steve Jobs was a hippie. And aside from being brilliant, he believed in, initially, about, he didn't want to undergo surgery. He had pancreatic cancer. He actually had a version of pancreatic cancer that is actually not as aggressive as most pancreatic cancers. So most pancreatic cancers kill people within a year of detection. He had a slow-growing pancreatic cancer, and he thought, well, I'll deal with it by changing my diet and doing all these kinds of naturalistic homeopathic remedies instead of going in and removing it. Unfortunately, it didn't work. And in the end, he lasted for quite a while with this cancer. It killed him. So he lived for like 7 years with the cancer. But if he had actually gone and had surgery and then regular chemotherapy, he probably would have survived, yeah.
So did he live those 7 years with it because of the things that he was doing, or just because it wasn't severe?
It wasn't severe, it wasn't, yeah. It wasn't especially aggressive cancer. So anytime, I don't know what kind of doctors you folks go to. Anytime it looks like you might have cancer, you should go to a real doctor. Okay? And not someone who's going to give you herbal treatments. They don't work. Okay. So one of the common ways to kill cancer is radiation therapy. So they direct a beam of X-rays or high-intensity X-rays onto the tumor. They're better at it now. So in the past, obviously that damages a lot of cells, including surrounding tissue. In fact, that's actually why it works. Because by damaging cancer cells, it makes the immune system recognize, oh, this is a damaged cell, it's time for it to go. So it creates damage, the immune system, tumor suppressor cells come along, or molecules, and apoptosis happens, and it's very effective. Now, the thing is, if there's actually mutations in the tumor suppressor system, and that's why there's an initial cancer, because it wasn't initially destroyed by the immune system, then usually radiation therapy will not work. Because it works by having cells show that they're damaged, and then the immune system comes along and destroys it with the tumor suppressor system. Okay. And then there's the standard cancer therapy, tried and true. Chemotherapy and the original chemotherapy agents are still used. Basically, what these agents do, these chemotherapy molecules, is they're toxic to everything. But they're generally more toxic to cancer cells than to normal cells. And so basically what you're doing with standard chemotherapy is you're trying to kill the cancer cells before it kills you, but you're going to oftentimes get very, very sick because of it, because it's damaging your own tissue. In fact, a lot of people actually, you have to get just the right amount of the chemotherapy. Not enough, you don't kill enough cancer cells. Too much, and you end up killing the patient. So, in other words, cancer therapy in this regard is oftentimes worse than the disease. It makes you really, really sick, your hair falls out, all that kind of stuff. It's brutal. Now, there is also something called targeted chemotherapy, where they have agents that work specifically on cancer cells and nothing else. Those are great. And sometimes what they do is they create a cocktail of a bunch of different chemotherapeutic agents so that it attacks the cancer cells on different parts of its life cycle. Now the trouble is, with targeted chemotherapy, is that, as I mentioned, cancer keeps evolving. New cells during replication phase, new mutations happen. Some of those mutations are beneficial to the cancer. And so these targeted chemotherapeutic agents, there might be a new mutation that they don't work on. So you might have initially a lot of success. It kills, let's say, 95% of the cancer cells. But as time goes on, mutations build up, and those cancer cells are then immune to the targeted chemotherapy. Just like what happens with antibiotic resistance. Yeah.
Because I normally hear about chemotherapy more than the radiation.
Mm-hmm.
So is it that this is like cheaper than radiation, or?
No, it attacks different. So radiation, what it does is damage the cells so the immune system comes along and kills it. Chemotherapy actually kills the cells directly by interrupting. In some cases, it stops it from the cancer from creating new blood vessels. In other cases, it interrupts the cell cycle in terms of replication, so it works differently. Good question. Now, one of the reasons, one way to get around that is to use a cocktail that attacks cancer cells in multiple different ways. So if there's a mutation that's likely to make the cancer immune to one chemotherapeutic agent over time, it's unlikely that there'll be all these other mutations that happen for all the different chemotherapeutic drugs that they actually use. So that's what they do, in fact, in terms with AIDS. AIDS is not cancer, obviously. But initially, when they were treating AIDS patients, they gave them one drug, an antiviral drug called AZT. It worked initially, but then over time, there were mutations in the virus that made it not work anymore. Well, now they use a cocktail of 3 different drugs. And so that's why there's a lot of people with HIV that are walking around and they're surviving just fine. It keeps the virus in check. Well, the same thing will happen with a cocktail of different drugs that attack cancer cells in multiple different ways during the cell cycle. Okay. So, standard chemotherapy, it kills all kinds of different cells. That's why people who oftentimes are put on standard chemotherapeutic agents, they have hair loss, they have constant nausea, they also have a suppressed immune system, and so they're oftentimes, they get secondary infections very easily from microbial pathogens. Okay. So, Herceptin for breast cancer specifically blocks the function of an oncogene called HER2. It doesn't harm normal cells. There's very few side effects. Unfortunately, as evolution happens in terms of new cells and new mutations, it oftentimes stops working after a certain period of time. Okay. Now, you've probably heard about immunotherapy. That means, as I mentioned, cancer has immunosuppressive functions that protect cancer cells from the immune system, that basically suppress the immune system so the immune system doesn't attack cancer cells. In fact, our whole body, all our cells have those kinds of things because otherwise our body's immune system would be attacking our own cells. In fact, there's a lot of diseases that happen precisely because of that. They're called autoimmune diseases. There's a slew of them: multiple sclerosis, rheumatoid arthritis, Crohn's disease, all kinds of different ones. So, these new treatments are designed to get the immune system to actually attack the cancer cells, to overcome that immunosuppressive system that stops that from happening. One of the first kinds of immunotherapy agents that were used were called checkpoint inhibitors. Remember what we were talking about checkpoints during the cell cycle. There's one during G1 phase, there's another at the end of G2 before we go into mitosis, and then during mitosis there's another checkpoint inhibitor. So some cancers, like melanoma, it was found that checkpoint inhibitor drugs actually do a very good job, and there was a large extension of survival time. So initially, from the time that, let's say, melanoma was detected as stage 4, usually within the first year, the patient was dead. Well, these checkpoint inhibitors, now they're actually extending life up to a couple of years, which is a tremendous amount when you think about cancer therapy. In fact, it's actually, some of those treatments, for some patients, they actually are completely cured of the cancer. The trouble is that there are some patients that would get really, really sick because it turns out the checkpoint inhibitors were now causing an autoimmune disease for the patient's own cells. So one of the recent discoveries was patients who were getting chemotherapy, it destroyed all their islet cells that produce insulin in their pancreas. So suddenly, it's like they had type 1 diabetes. They had to take insulin from then on. It's kind of a trade-off, but it's pretty brutal, and it could be other kinds of tissues as well. And so they're realizing that checkpoint inhibitors aren't necessarily as specific as they'd like to be. But there's other ones, there's other therapies. One of them is actually a vaccine, a cancer vaccine. So normally what we do with a vaccine for, let's say, a viral disease is we either take a dead virus or a weakened virus, or we actually make a vaccine out of certain proteins on the virus that the body recognizes as non-self, and the body will then attack that virus. Well, they're doing the same sort of thing with cancer cells. They're taking certain aspects, proteins on the cancer cells that normal cells don't make. They're then creating a vaccine, and they're getting kind of like an antibiotic effect or an antiviral effect. In this case, this is against your own specific cells. So some of these newer immunotherapy agents are showing huge effects in terms of actually curing people, where suddenly their tumors shrink and they're cured, or it keeps the tumor shrunk for a long time until there's new mutations, and then it suddenly stops working again. So there's a lot better, well, there's more hope than there used to be for a lot of cancers, where people are suddenly completely cured of a cancer that was, before that, was stage 4, metastatic all over the body. There was no way you can go in and surgically remove all these different tumors, but you have this agent that's actually killing them all in place. So, like I said, there's checkpoint inhibitors, monoclonal antibodies. You don't have to remember all these vaccines, things like that. Now, in terms of cancer therapy, whatever it is, whether it's standard chemotherapy, targeted chemotherapy, immunotherapy, radiation, whatever, oftentimes what will happen is cancer will go into remission. In other words, the tumors will shrink, it will stop spreading, the symptoms that the patient is suffering from will disappear. A remission can happen or can occur for maybe just a year, or it could happen for a long time. So usually if cancer doesn't, after it's been treated, doesn't show up again beyond 5 years, you're considered cured. So this is, let's see, Patrick Swayze, I guess. I think he had pancreatic cancer. In the end, it killed him. Okay. So one of the reasons pancreatic cancer is so deadly is usually it's not caught. It doesn't show symptoms until it's really advanced. The same thing happens with ovarian cancer. So ovarian cancer for women is really a deadly disease. It's oftentimes at stage 4 before it's actually recognized and detected. Now, prostate cancer for men usually shows symptoms earlier because the man as a prostate cancer tumor expands, it causes urinary problems. And so oftentimes the man will start having difficulty peeing, completely avoiding his bladder, getting up many times during the night. He'll then go in for a PSA test or something, and it's detected fairly early. Also, it's a lot of prostate cancers are not as aggressive as ovarian tumors. Some are, though. So there are some prostate tumors that are very aggressive, just like kind of ovarian tumors. The trouble with, generally with ovarian cancer, they're getting better at it, but early detection, there's no early detection test like there is for prostate cancer with a PSA test. There's another thing. There are oftentimes tumors, cancerous tumors, that are never going to be a threat. They are localized, they don't metastasize, they grow very slowly. So like a lot of prostate cancers, they grow so slowly that usually they don't go in and remove them, because eventually the person who has them is probably going to die of something else from old age, right? There's also breast tumors, breast cancer tumors that are like that. So one of the problems with early detection is detecting cancer that actually isn't dangerous, that if it was not surgically removed, it would not cause any problems. And so you're basically treating a cancer with invasive surgery or chemotherapy or something like that that's likely to cause harm to the patients when you don't really need it. So for a while they were discovering all these very small ductal milk duct breast cancers, very, very small. And they were removing them all. And then they realized that, that a lot of these cancers were not going to really grow beyond what they already were, and that just monitoring them over time was a better option than actually going in and doing surgery on these tumors. So ovarian cancer is often metastatic. It gets into the lymph system, which is part of the immune system. It can travel all over the body. And so you end up with cancer everywhere. Okay. Has anybody ever heard of HPV?
Yes.
Okay. So HPV is a virus. Human papillomavirus, HPV. It's a sexually transmitted virus. It causes genital warts. It can also cause an infection in, if you have oral sex, in the mouth and the throat. It was responsible for about 99% of all cervical cancers. They now have a vaccine that protects people, and so they're suggesting that especially women from about 9 years old to 12 to get vaccinated before they start having sex. And also guys, too, because guys can get infected with this virus as well. That's why it's sexually transmitted. Unfortunately, and it does it, it has two genes, the the HPV virus that can each inhibit a different tumor suppressor system. So in other words, it's a virus that can suppress the immune system, and that's the actual tumor suppressor genes. It turns them off, and then tumors eventually show up. It's oftentimes after infection, it might be 20 years later when this happens. The incidence of cervical cancers now has gone way down. First of all, it's easily detectable with pap smears and regular gynecological exams. And so the number of people getting cervical cancer has gone way down because of early detection. Unfortunately, a lot of Americans are, they're crazy about sex. In other words, they're crazy that they don't want their kids having sex. And so a lot of actually religious people are saying, I don't want my daughters vaccinated, because then what I'm saying is, oh, if I get you vaccinated, that means you're going to have sex. And it's okay to have sex, and I don't want you to have sex. And so, of course, eventually, their daughters have sex anyway. They get cervical cancer, and then they die. So the HPV vaccine is actually really underutilized. Only, I don't know, like 50% of girls that are early teenage girls are actually vaccinated, and boys even less so. Okay. So, as I mentioned, a lot of religious people argue that government-sanctioned administration of the vaccine is immoral. There was a governor of Texas, Rick Perry, and even though he's very religious and conservative, he wanted everyone to actually get vaccinated. But he received a lot of pushback from Southern Baptists and various other denominations in Texas. Okay. So remember, there's two kinds of, there's somatic cells. Those are all of our other cells besides germline cells. Germline cells only create sperm or eggs, depending on if you're male and female. All other cells, most cancer is in somatic cells or autosomes. Autosomes are not heritable. In other words, you're not going to pass a mutation on if you develop, let's say, a mutation that causes cancer. You're not going to pass it on to your offspring if it's in an autosome or somatic cell. That's only going to happen if it's actually a germline cell.
What about sickle cell, I believe? Can you pass that?
Oh yeah, yeah. But that's actually a genetic heritable disease. And we'll get into that more when we talk about pathogens. It turns out it's a recessive disease, so you need two bad copies for patients to show sickle cell disease. Whereas if you have one bad copy and one good, in areas where there's lots of malaria, it actually protects those people from getting full-blown malaria. Okay, so in terms of germline, the only way things are heritable, like sickle cell, is actually through mutations that are passed from one generation to the next through the sperm or the eggs. Okay. Now, one example of a heritable cancer is called retinoblastoma. Now, occasionally you get sporadic retinoblastoma. So you end up with, let's say, a mutation happening. Okay, so that's one mutation. It's not going to cause retinoblastoma. But then the possibility that a second mutation will occur in the other chromosome. Remember homologous chromosomes. And so you can actually end up with what's called sporadic non-hereditary retinoblastoma. Retinoblastomas create a tumor in the eye. But there is a hereditary form of retinoblastoma. So someone inherits one bad copy, and then you end up with another that happens sporadically, and so it's much higher incidence of getting actually retinoblastoma tumors. And the hereditary form usually means that if you get it in one eye, you're going to get it in the other. Whereas a sporadic form, you're usually only going to get it in one eye. Standard therapy for retinoblastoma is to remove the eye. For the hereditary form, it usually shows up early in a child's life. I think it's actually a sex-linked trait. And so, so there's this fairly well-known fellow who's blind who can actually ride a bike. He doesn't have any eyes, and he's got, fake eyes. And he uses something called, well, he uses like what bats use in terms of sonar. He makes these clicks. We'll get into that later. I'll show you. His name is Daniel Tish, and he's amazing. And he somehow managed to teach himself how to use this to actually get around, just like bats do, whether it's dark or light out. He obviously can't see. So retinoblastoma, the hereditary form, it's got something that's called penetrance. That means it's got 90% penetrance. That means that if you have the mutation, there's a 90% chance that the individual is going to get retinoblastoma in both eyes. So very high incidence compared to the BRCA tumor suppressor gene that leads to breast cancers. That has penetrance that varies between 20 and 80%. Okay, so here's the BRCA. A lot of women who inherit this mutation in the tumor suppressor gene, they have a much higher incidence of cancer, and a lot of them opt for having their breast removed and then reconstructed. Angelina Jolie. Others decide not to, but they have increased monitoring for breast cancers. BRCA also increases the odds of ovarian cancer and prostate cancer in men. So you can read this. Okay, good.